Hello, everybody. Thank you for that great introduction. Thank you so much for being here. I'm hoping that over the next hour or so, we can just have a bit of fun and learn a little bit or a lot about the big question of, is diabetes getting on your nerves? So maybe you already kind of mentioned my background, but I just wanted to let you know like who I am, how I got to where I am. So after my degree at Durham, I started as a lab technician at Newcastle University. This is my first role. And it's kind of funny, I've kind of come full circle because I sort of have sensory pain neurons here and then sensory pain neurons here and then everything in between. But we published a paper on how to develop these neurons from a population of stem cells you find in the skin. And then after this, I then worked in a startup company and did a lot of reprogramming and a lot of differentiation. If you don't know what I'm on about, don't worry, you will by the end of today. Then I came from Newcastle to London to work in this brilliant research building under Dr. Bochukova. And we performed a lot of work on the genetic of obesity and the hypothalamus. If you are curious about that, don't hesitate to catch me at the end. But now I work in the Clark group, Alex Clark, and we are interested in sensory neurons and diabetic neuropathy, and that's what I'm going to focus on today. Okay then, so what is diabetes? Let's just look first at a healthy individual. So you can see this person's had a cheeky Nando's. And after you've eaten food intake, this increases your blood glucose sugar levels. The pancreas then detects this, produces insulin, and then this insulin travels around the body and tells cells, use up this glucose, we've got it, let's do it. And then this returns the blood glucose to normal levels. Now, obviously, I can't talk about diabetes without talking about type 1 and type 2. We all are probably very aware of this. So type 1 diabetes is actually an autoimmune disease, whereas type 2 diabetes is a metabolic disorder that develops over time. So how does this affect that blood glucose pathway? So first, for a type 1 diabetic, cheeky Nando's, blood glucose levels have gone up. And now the pancreas is unable to perform its function because immune cells, there's a really angry looking one, attacks the pancreas, and therefore it's not able to produce insulin, so the blood glucose levels remain uncontrolled. Now for a type 2 diabetic, it's slightly different. Cheeky Nando's, blood glucose goes up, but this, because it tends to happen over time, it can affect you in two ways. So the first is that the pancreas kind of gets worn out and it stops making insulin. But then on top of that, the cells that are supposed to respond to it don't do so, and therefore, ultimately, same effect, you're not regulating your blood glucose levels. It's a big problem. That's why we are researching it. You can see here this study in The Lancet estimated at the time of publication that around 6% of the global population have type 2 diabetes, which is obviously a vast number of people. And this is predicted to keep rising and rising and rising. I mean, you can see by 2050, over 10%. And there's a lot of reasons why this might be the case. But the major ones are, of course, increased levels of obesity, which I guess go hand in hand with also access to high calorific foods and sedentary lifestyle. But then also we are just, as a global population, getting older, living for longer. And also quite a grim one is just people are more stressed. So there's that as well. So that's diabetes, but we're actually interested in diabetic neuropathies. So neuropathy is just the fancy word for when your pain-sensing neurons aren't functioning properly, and then this causes problems with patients. So diabetes is actually the leading cause of neuropathy worldwide. And of those, already high percentage of people in the world that have diabetes, 30 to 40% of these are reported to develop neuropathic pain. It is the leading it's the leading cause of diabetic patients self-reporting themselves to the NHS. So it's the main reason why diabetic patients go to a doctor and say, I'm uncomfortable, I'm not doing well. And there are different kinds of symptoms that kind of come under the neuropathic umbrella. So it's not always pain, but it can be numbness, weakness, pins and needles. And I think it's worth noting at this point that it affects mostly the periphery, so the hands and the feet. And the image I'm showing here is a common test where a monofilament wire is used on the foot of patients, and it's basically a way to determine whether or not there's insensitivity or oversensitivity, etc. So why do these diabetic patients develop neuropathies? So first, I think we need to understand the basics of how a human responds to pain. So what you have is sensory neurons in the skin, and they can then create a response when they are exerted by mechanical, thermal, or chemical damage. So a hot pan, if you touch a hot pan, you can feel it, right? So the sensory neuron's job is to then take that information from the periphery to the spinal cord, which then takes it to the brain. The brain now processes these signals as pain, and then this has many functions, including a protective response. You want to be able to flinch away from the hot pan. And the main leading hypothesis, which is a bit problematic, and you'll see why in a second, is that basically diabetics have high glucose, and then this causes damage to the neurons, which means that they don't function properly. And this has been proven in most cases. And what are the treatments for diabetic neuropathy? So because of the lack of real understanding of the mechanisms that are at play here, they're severely lacking, which is one of the main reasons we're doing what we're doing, and I'll get onto that in a moment. But pharmacologically first, there are different ways that they attempt to alleviate the symptoms. Antidepressants, a lot of the pain happens at night, so this can help patients to fall asleep, etc., etc. Painkillers, and I think we can all imagine how problematic that is due to the addictive nature of opioids and things like that. It's also not dealing with the underlying issue, it's just dealing with the pain itself. And then anticonvulsants are a type of drug that just kind of reduce the activity of these sensory neurons. Then we've got a bunch of other things. So we've got diet and exercise, which again you're going to see might not be ideal for everybody. Physical therapy, so increasing circulation and muscle strength and stuff like that. And then a last resort, as always, is surgical intervention. And in worst case scenarios, these kinds of conditions can actually lead to amputations. But this is the real head scratcher, and why we can't wait to get into this problem and figure out what's going on, because there is actually no correlation between how well diabetes is managed, so how the glucose levels are maintained, and the pain that these patients ultimately report. So if we go back to this slide, telling someone, oh, we'll just manage your glucose levels, it's not enough. There's something else going on. So one of the first point of call whenever investigating any scientific problem is to kind of look at what we already know. And I really want to just mention this HSN1 neuropathy to you now, because I'm going to be showing you some data later that's related to this, because our diabetic work is very preliminary at this stage. So one clever way is to just look at known neuropathies. I don't want you to overthink this, but basically this is it's very rare, but very severe. And we know exactly what causes it. There's a mutation in one gene, and then the patients have this abnormal sensory neuron behavior, and a lot of the symptoms overlap with that of diabetic neuropathy. So in summary, we seek to find relationships between this less common severe neuropathy and the diabetic neuropathy, which, if you remember, is the leading cause of neuropathy in the world. How many of you guys have actually been in a working laboratory? Not the people that work here, no. The people that don't work here. Yeah, anyone? Yes? Nice, brilliant. So yeah, not a lot of hands. I always remember, even in undergrad, sort of seeing lectures and talks and being like, it's really hard to picture it. So what I'm trying to achieve today is to kind of really give you a feeling for how you can do what I do. And there is an activity at the back that we can play with later where you can kind of make your own neurons. So we'll see how that goes. Okay, so this is what I do. This is how I'm going to tackle the problem. I'm going to use human-induced pluripotent stem cells, which from now on I'll call HIPSC because it's easier to say. And the thing about studying things like neuropathies or disorders of the brain or disorders of other peripheral neuronal things is, unfortunately, you can't go around chopping people up and looking inside. So yeah, you can't really go to a diabetic patient and really take a look at what's going on in the arm. That's not good or ethical or whatever. So let's just go to the basics on what is a stem cell. So we start as human beings as just two cells, a sperm and an egg, and ultimately this develops into an embryo that ultimately develops into a person, a baby. And this thing has brain cells, eye cells, heart cells, muscle cells, you name it, and pain-detecting sensory neurons, which is what we're interested in. So researchers have found a way to harness this technology, but in the dish. So that was embryonic stem cells, but what I talk about, we don't actually use any embryos. And one of the main problems with studying an adult that has diabetes is obviously the cells that they have can no longer become anything. So there's this very famous paper from Yamanaka and colleagues that basically developed a way to take an adult cell, something that's easily accessible. So I could take 8 milliliters of your blood, I could take a skin biopsy from you, and then over a period of a few months, I'm able to then turn those adult cells into a stem cell. And the thing that's so amazing about these cells is they retain the footprint of the donor. So if you have diabetes, those stem cells that I make from you via your adult cells will have diabetes. And I don't want you to think small. We're talking about diabetes today, but this can be used for like any myriad of diseases. In this building, we've got people studying muscular dystrophy. So just exchange diabetic stem cells for muscular dystrophy. And it's just a really brilliant way to model disease. So I'm just going to explain to you like really briefly how do you get a stem cell from an adult cell. So a really common way is to take a bunch biopsy. It's only about this much skin. And then we're able to grow the cells in tissue flasks, just like the ones you'll see at the back. And then this is the part that's important. So even though a stem cell is super powerful, it's also kind of simple because it's like the first cell. So we can manufacture a virus that turns on the 4 pluripotency genes that make the cell a stem cell. And then ultimately we end up with this population of stem cells, either from healthy controls or diabetics, that can then be turned into theoretically any cell type in the body. So more specifically to us, this is our experimental pipeline. So, yep, as I just said, we've got the patients, we've got the fibroblasts, we've introduced the reprogramming factors, and we now have a stem cell that has different kinds of diabetes. So you have to always include a control because you've got to compare it, right? So these individuals, we try to match them as much as possible by like their gender or their age. So we've got those stem cells, and then we've got the type 1 diabetics as well, here at the bottom. And then remember what I was saying about this confusing exhibition of pain from the type 2. We've got a range of different kinds of patients. So we've got type 2 diabetic patients that have painful neuropathy, sharp pains, like really nasty stuff. Then we've got the painless neuropathy, so that's more to do with like pins and needles, tingling, weird sensations. And one I've missed out here is just type 2 diabetics without any form of neuropathy. And then what I do is, in unison, differentiate all those cells into the neurons of interest, the sensory neuron, the pain-sensing sensory neuron, and then from there we can do a myriad of cell phenotypic assays, and we're going to talk about some of those now. So how do you make a sensory neuron? I always joke it's kind of like a recipe. So fortunately, researchers have developed over the years different methods of developing different cells. But basically, the cells are grown on tissue culture flasks like this. They're, in this case, adherent, so they're stuck on the bottom. And then that red liquid is media, which is food for the cells. It has all the things a good cell needs, like salts, sugars, all the good stuff, and non-essential amino acids. And then what you do, basically, is change what's in that liquid. So the liquid's simple. That stays the same. That's this arrow at the top. But then if you can see all these crazy numbers and letters and stuff like that, these are special molecules that basically just copy of what happens in the embryo. So we're just mimicking how the very few cells become the full-grown baby in utero. And then you have to mature them for a very long time for them to become active. So these are my stem cells, but imagine I've got control, diabetic, 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 control, control, and then they grow, I don't know if you can see very well, but they grow in like these colony-like shapes. And then after doing this recipe, we end up with these beautiful neurons. So I think you can see, so these clusters here are like a cluster of cell bodies, and then the lines coming out are the neurites. If you don't know what that is, don't worry, we're going to get onto that. And I always like to include this photograph of me because I think it's good to picture what I'm actually doing. So this thing here that I'm up against is a sterile tissue culture hood. So that it blows sterile air up towards me, because this environment is also really nice for bacteria. So we're trying to keep those out. And then I perform work here in a sterile environment. And then if you can see this thing behind me, this is an incubator. So it keeps the cells at 37 degrees, 5% CO2, just like in the body. And then I just wanted to talk about this really nifty little method because this protocol only creates a certain percentage of neurons. There's a lot of commonalities in the journey to becoming a neuron, so we're going to get lots of other kinds of neurons in there that aren't the pain neurons that we're interested in. And I always like to show this because I just think it's so cool that you can stick magnetic beads to the cells and they still survive. It's a very tactile thing that we can do. So bear with me. These are these cells floating around there in the tube. So I've detached them and singularized them. And then inside there, we've got the small green ball, which is a sensory neuron, a cell that isn't a sensory neuron, so it's negative for the sensory neuron marker. And then the little red cell, the little red circle, is sensory neurons that are sensory neurons, they're positive for the marker. And then the big grey ball is a magnetic bead that sticks only to the sensory neurons. So we allow those to kind of hang out together for a while. And then what we do is we pass the cells through a column. And then can you see this massive black thing? It's about this big. And we let the cells pass through the column. And this is a huge magnet. So it holds on to the cells that have this magnetic bead associated with it. And then once we've allowed that to pass through, we then put the column into a tube and we collect we push it through, so now we're getting the magnetic stuff. And I think you can see here, well, not so good, but you can see here that without the saw, we've got these beautiful bright circles with the neurites coming off, but then a lot of other stuff as well. But when we do it this way, you actually get this really nice population. So I thought that was a nifty little technique you might like to see. Okay, so now I'm going to spend some time going through the different kinds of analysis that we can do on our sensory neurons. So a really, really common step that you'll probably will come across if you're interested in any of this stuff is something called immunocytochemistry. I'll explain how to do that in a second, but basically these neurons here are these neurons here. So just way more zoomed in. So this circle here is one of these small circles here. And what this is showing is the nucleus of the cell and then this green stuff. And the green stuff is the structure of the neuron. It allows us to visualize because it's the stuff that supports neurons. So in its most simple form, basically, we just add a primary antibody that is designed to stick to the thing we're interested in. and then we add a secondary antibody that sticks to that, and then that will then glow and make all these beautiful pictures for us that can be visualized as long as we have a special microscope. So again, so if I just explain to you again how the actual structure of a neuron, and that will help you in the slides coming up. But basically, we have the cell body and then axons that come off it. And these are very important for the function of the neuron. So remember, I wanted you to keep that HSN1 in mind. I want you to imagine we're going to repeat this, but for our diabetic neurons, and see if we can see a difference. But you can see the bright green, which is that structural element in the healthy neurons. And then you can see the bright green in the ones with the neuropathy. And just straight away with your eyes, you can tell there's something very wrong here. The neurites aren't as long. And then this is an example of our quantification. So we can measure the length in microns. How long are these? How much do they branch? How complex are they? And this is just one way to kind of uncover the difference between disease neurons, sensory neurons, and controls. And then immunocytochemistry can also be used. It doesn't have to be used for the structure of the neurons. It can also be used to stain for things that are important for the function. So by nature, neurons like to make synapses so that they can send signals to each other. And I always really like this pretty image here, where you can you see those green dots, like dot, 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 all the way along the axon? So this is a postsynaptic protein. It's responsible for that part of sending signals. And what we can do is we can count the density of the puncti along each measurement, and then ultimately, again, get a phenotype, get something that's different that can be targeted to help alleviate the symptoms. So this is, it's not for diabetes again, but it is for another disease phenotype. And you can see, again, in the controls, there's more of this protein, but in the disease, there's less. And you can't talk about neurons without talking about neuronal activity. So they like to talk to each other, they like to send signals, and they do this by way of electrical activity. So we're very proud of this new toy that we have here in the Blizard. It's called a multi-electrode array, or a MAIR. And basically, you grow the cells in these very funky and expensive little dishes. And what you can do is you can look at the electrical activity of a larger population of neurons. And something that I always like to bring up here is that because these guys are sensory neurons, they do actually respond to capsaicin, which is the thing found in chili pepper. So just like we do, like we sense, our cells actually do that too. We ordered like fancy lab capsaicin. We don't like mush up peppers and stuff, but it does make them fire. So you can see here, voltage being a measure of the activity, that there's not so much going on, a little bit, but then after you add the capsaicin, they light up. And then, yes, my video worked. So this is another way of measuring activity. Don't worry too much about the calcium, but again, this is a fluorescent dye that labels the calcium. When you see that light up, that's activity of the neurons. And again, this is an example where we've added the capsaicin. And there we go. And another thing, so what we could do now is we could do that with diabetics and controls at the same time, and maybe see if there's a difference in the activity of these neurons. So this is actually pretty cool. So researchers have developed a conjugate. That means like it naturally has something that glows stuck to it. So tetanus toxin. So tetanus actually hijacks the axonal transport and travels from the periphery to the dorsal ganglion. And again, this HSN1, it's back, so we're definitely going to repeat this for the diabetes. And you can see like this beautiful transport shuttling along from the healthy controls, but then you can see something's not quite right here in the monogenic neuropathy case. You can see that the list goes on, right? You can do whatever your heart desires, and we just try to be smart about what we choose based on what we think is wrong with diabetics, neuropathies. This is a cool one where we grow a monolayer of neurons and then we scratch them, and then we video them over 48 hours, and you can assess the rate at which the neurons are able to recover the gap. I think, yeah, and it can assess movement, but also division, and it's also a measurement of how well the neurons cope with being injured. Okay, so that was a lot of assays, but I just really wanted to show you the potential of these models and all the different things that we can do and are going to do to try and find out some novel things about diabetic neuropathies. So what does this actually mean for diabetics? So just to remind you, we've got these iPSC-derived sensory neurons. We've got them from diabetics with and without painful and painless neuropathy and controls. And then we're just going to do a huge range of analyses that no one's ever done before in this environment, on these kinds of cells. And ultimately, we're trying to develop a greater understanding of diabetic neuropathy in general, but also about why, if you're regulating your blood glucose the best anyone's ever done, why do you still have more pain than someone else? So obviously this is going to lead to treatments. If we understand the mechanisms behind what's going on, we can introduce drugs, we can introduce other forms of treatment. But it's also important for diagnostics. We might be able to, in the future, analyze you in some way and then be able to predict if you're going to suffer from neuropathy, like what is going on. And that is our goal. So I just want to thank everybody. Thank you for being here. Thank you to Centre of the Cell for having me. This is my group, Alex and Alberto. So yeah, we've been working together for nearly a year now, having a great time. And I would really, really, really like to just mention and thank the patients and their families. All of those type 2 diabetic lines come from patients who've donated their skin biopsies for this study. And yeah, we really hope that we're going to be able to help them. Thank you.